Good morning. Greetings and salutations. My name is Jaidyn Thomas, and I am a recent graduate of the Jackson State University with a bachelor's degree in computer science. Today, I actually had the opportunity to spend some time in New Hampshire, at the University of New Hampshire, quantifying the relationship between clearance rate and valve gaping in Eastern oysters. And today, that's what I'd like to discuss with you all, right? So let's get into it. Eastern oysters are foundational species. Now, you may be asking, what is a foundational species? Well, a foundational species is defined as a critical species that shapes the structure of a community. Examples, you have your coral, right? You have your bivalves, which are your oysters, mussels, things of that nature. You also have seagrass, and you have like really cool oyster reefs. Now, you may be wondering, what's the importance of foundational species? Well, they play a crucial role in the actual structure and maintaining the habitat, right? They do this by creating and maintaining, enhancing the biodiversity, as well as contributing to the overall health and stability of the ecosystem. Now, specifically, oysters are very important because they filter, right? We're going to get into that, okay? Eastern oysters, a little bit about them. You typically find them along the coast. This specific type, you find them along the East Coast, all the way up to the Gulf of St. Lawrence, all the way down to the Gulf of Mississippi, or excuse me, the Gulf of Mexico, pardon me. Their habitat, you find them in brackish and salty waters, 8 to 35 inches, 8 to 35 feet deep, and you typically see them in their intertidal and their subtidal zones, which means that they're kind of on the coastline to a little bit deeper in the water, right? This is what an oyster looks like. Aren't they cute? They just look so adorable, right? Great. So they average about 3 to 5 inches, at the most 8. Their lifespan is about 20 years, and you typically can figure out their lifespan by the rings. So similar to a tree. If you look at the rings on like the little shell, you can figure out their age. What's very important about them is that when they are adults, they are considered sessile, which are stationary. This is important. That means they don't move, right? So if they don't move, what do they do? Okay, we're going to get into that. Human wise, well, for the humans, not only are they important to the actual ecosystem, but the economy, it's a huge economy, right? They bring in about 8 billion annually through aquaculture, through oyster farms, it's through tourism, through jewelry. Baby, you can do everything with oyster, okay? Right? So let's really get into it. Oysters, because they are sessile, okay, they do this one behavioral feature, which is called valve gaping. All right. You may ask, what is valve gaping? Well, it's the opening and closing of their mouth or their hinge. Okay. That's all they do because they don't do. Right. This is very important because this is for their feeding. This is for their spawning. This is for filtering water. This is their main behavioral feature. Now, in relation, we discuss clearance rate. You may be asking, what is clearance rate? Well, if you look at this diagram, clearance rate is defined as the amount of water that, or the process of them filtering the water through their system to filter out the particles, okay? Now, this is important because it can keep the nutrient cycle kind of running through the ocean, keep the algae down, that kind of thing. So if you think about that, that's kind of where we're leading with the oysters. With this, we tend to look at and say, when people have done research on oysters using valve gaping, there has been an assumed relationship between the two, but there hasn't been research done to confirm it. So I did the research, right? I sat there and I was like, let's figure out, okay, is there an actual relationship between the two? Which brings us to the research question. Is there a relationship? All right, let's find out. How do we go about doing this? I'm glad y'all asked. Okay. Y'all didn't ask, but I'm glad that y'all asked. All right. You gonna go? What? Hello? You go. Okay, we just gonna click. That's fine. That's fine. Okay, cool. All right. So to measure valve gaping, there's a thing called a Hall biosensor. Okay. What this sensor does is you take the sensor, you glue it to the bottom, and then you put a magnet on the top, right? And what this does is it collects the data of them kind of doing their opening and closing, and it measures the opening and closing movement over time. And you connect those sensors. The sensors are connected to a data logger, and you just continue from there, right? So we have a picture of what that sensor data looks like. We have the alive and we have a dead. When they're dead, they become, well, their mouth becomes open. So you obviously see that their line is a little bit higher. Let's zoom in and let's say here. All right. When you're looking at the sensor data, it's supposed to have your open and your close. All right. When it's open, the line is going to be high. When it's closed, the line's low. Pretty self-explanatory. Now, simultaneously, to continue and to run the experiment, right, you want to take and you want to measure valve gaping and clearance rate together. So to measure clearance rate is typically done in the lab. Obviously, because you can't take the water in the field and kind of contaminate it and be like, oh, okay, I'm going to measure this amount of water. You can't do that, right? So you have to do it in the lab. So to do that, we set up this cute little structure and I made them cute little homes. And so I put them in separate containers. I had 8 oysters total. And what we did was we took the oysters, we attached the Hall sensors and placed an air stone inside, and we added a concentrated amount of the species Rhodomonas algae. Now, over a 2-hour period in 40-minute increments, we were able to take the samples and collect the samples and just kind of collect the clearance rate samples to see how much, you know, they cleared over time, right? Here's a cute picture of what it actually looks like. Isn't that so cute? So adorable. Okay, they were very happy. Now, to process these samples, I put them in 96 well plates. Okay, I took 3 samples of each for each time period for each oyster, and using a flow cytometer, which basically is a really cool instrument that counts the amount of cells in that concentration. And we, once the flow cytometer did its work, I took the calculations and I averaged them out. Now, you may be thinking, no, Jaidyn, what did you figure out? Well, here we go. Here is our clearance rate. Now, as you can see, we have 4 oysters that ate, which are signified by this lovely little circle right here. And then we have 4 oysters that did not eat. Okay, if you specifically look in the 40-minute time period, you can see that there is a clear difference, and it's a very distinct difference that the 4 oysters that ate, you see a depletion in the cells that were in that sample and in the water, right? Pretty self-explanatory. It's kind of cool. Now, the oysters that didn't eat, we'll get into that later. Now, let's compare it with the sensor data. Oh, I'm sorry. So when you really look at it, the feeding oysters averaged about 500 cells and 500 cells per milliliter. And the non-feeding oysters, they averaged about 50 cells. So you can clearly see that there's a huge difference. Let's look at the sensors. Okay. I know this looks confusing. We're going to break it down, right? So up top, we have feeding. On the bottom, we have not feeding. Okay. The first indication is when we place the oysters in the experiment. Second indication is going to be when we started the experiment. And the 3rd through 5th lines that you see dotted are going to be the times points that we were taking everything. So when you really look at everything, you can clearly see, based on the diagram that I showed you all earlier of the alive versus dead, that there's a distinct movement and that there was a distinct gaping measure, an opening or closing, right? Those are the 4 oysters that ate. We love them. Okay. Now let's get to the ones that didn't eat. Let's break it down. Let's start with J5 and J6, which are going to be these 2 right here. They, if you noticed, and refer back to what I said earlier, with the line being on the bottom, they're closed, so they didn't really eat. So obviously, they're going to be towards the bottom. Cool. J8, on the other hand, is kind of interesting. He was open the entire time, but you don't really see that movement of them actually opening and closing and kind of either breathing or signifying that like he was doing something. So he just kind of, I don't know, he just kind of sat there and he was, I don't know, he might have been singing a song or something, whatever. J1, which is really interesting, we're not really sure about. It may have been a sensor error or he may have been a little wonky, but as you can clearly tell, it does not look like the rest of them. So I think that could be something that I could do in the future, kind of figure out. All of this is important because when you're wanting to use, oh, I'm sorry, I went too fast. All of this is important because when you're wanting to use and measure valve gaping and clearance together, valve gaping is typically done in the field. But clearance rate, like I said, is done in the laboratory setting. And if you're wanting to see how much they've possibly filtered, then you need to, the goal is to try and use your valve gaping data to assess how much they filter over time. What did we find out? Well, we found out that there's a clear difference between the oysters that eat and the oysters that do not eat, and you can clearly see that. We ran out a little bit of time, but what I'd like to do in the future is actually to take the two previous slides and the two graphs and kind of model them together and hopefully be able to create an equation that'll say if you take the weight of the oyster, the age of the oyster, the valve gaping measurements, and you should be able to get an estimate of a clearance rate to see, okay, how much could they be clearing over a certain amount of time? So this is kind of future work that I'd like to do. I think that these are my references. And I'd really like to say special thanks to everybody, especially a special thanks to my wonderful mentors back at home, my family, OECI. Initially, I really enjoy everything. Now we're going to get into the pictures, right? Okay, so cool, right? So this first little picture on the bottom, that's me collecting my little oysters. Don't I just look so excited? I love them. During my time in New Hampshire, I actually spent a little bit of time working at a oyster restoration barn. And so I was helping them with doing like oyster restoration, and it was really cool. So that's actually me in the second picture with all the little oysters, transporting them to like their new little home. It's really neat. Um, this is me on a little island. I have a thing for long socks. Okay. I just love long socks. Not everybody likes it, but it's great. And then this is me and my mentor. And this is my presentation. If we have any questions, I am open to them. Yes, ma'am. Of course.
Really interesting stuff. And I'm wondering, um, you, yes, thank you. So, am I on? Okay, thank you. So, I love the idea of the gaping, and I'm wondering, you said it does it for several different behaviors.
Yes, ma'am.
So, is there a way that you could use video or something along those lines to determine if the gaping was related to feeding, just to add that piece to it? And then, is there any indication that there's a difference in how wide it opens?
Yes.
And how that might infer behavior?
So to answer the first part of the wonderful question, to answer the first part, there is, I have known that there have been recent research in trying to actually video them over time. That is something that I would like to do because
Cool.
I kind of realized that I really like working with oysters. And so that could be something that I could add because I want to continue this. And so that I would like to measure them over time to see like how wide and everything. To answer the second part, there is you can do to see how wide. That was one part of the experiment that we weren't able to fully get together. We did start to technically to get like the actual measurements. You have to sadly mutilate them. But there is a voltage range that you can do to say, okay, if it's open this wide, it might be doing this. If it's open this wide, it might be doing that.
Thank you so much. Very good. Did you find a pearl?
I wish I did. Also, what I learned is that pearls are, if an oyster produces a pearl, it's actually like a virus, or it's not good for an oyster to produce a pearl. So I was like, oh, that's interesting. That's interesting. I learned a lot about oysters, y'all. I just like to eat them. So, raw Rockefeller, it was great. Sadly, and I'm so upset, I did not get a chance to actually eat an oyster while I was up there. And I that hurt my feelings so bad. Oh, that hurt my feelings, y'all. Does anybody have another question? Don't raise your hand all at once.
We have a question in the virtual chat. Can you all hear me in the room?
Oh my gosh, yes.
Okay, well, first of all, I want to say fantastic job. That was an amazing presentation.
Thank
And we have a
you.
question from one of our virtual attendees. What were some of the difficulties in collecting your data?
Ooh, ooh, oh my God. So the clearance rate was actually not the difficult part. It was actually the sensors. And what was difficult about it was part of the time, some of the sensors worked. The dead oyster that is actually on there, on the oh well. The dead oyster that I displayed was actually one of the oysters that we were going to use for the experiment. And his data came out looking weird. I think some of the sensors kind of got like waterlogged. So when we were trying to what we were wanting to do is with our valve, our voltage rating for the valve gaping, because it reads in voltage, we were trying to do it by an actual distance, like, okay, this is 1 centimeter wide, this is 2 centimeters wide. And so we were in the process of doing that, and we kind of figured out that something was wrong with the sensors towards the end, not the experiment, okay, just the end of the just that part. So I think the sensor was a little bit challenging, but yeah. Any more questions? If not, I can do a little jig. Yay!